Well, hello everyone. If you're new here, welcome. If not, welcome back. While we let other people trickle in, I encourage you to introduce yourself in the chat, maybe your name, state, occupation, because not everyone's teachers here. And if you're a teacher, please tell us what grade do you teach and anything else you would like to share. I would love to hear maybe what made you join the Nano Ed Forum today. While you introduce yourselves in the chat, I will introduce myself. Hello, my name is Maria Fernanda Campa, and I'm a staff scientist at the National Nanotechnology Coordination Office. I will be your moderator for today. If you're new here, the NanoEducators Forum is a collaboration between my office, Marielle Kolker, who's over there, physics and engineering teacher at Morristown High School in New Jersey, and Danielle Duran, Director of Education and Outreach at Nano at Stanford, and also a current high school teacher in California. The NanoEd Forum is a space for educators and nano enthusiasts to learn about the newest nanotechnology trends, share resource, lesson plans, and any other ideas on how to incorporate nano into your classroom curriculum in a fun and engaging way. The forum meets only 4 times a year, and we discuss topics of interest to the participants. So please let us know if you're interested in hearing about specific topics at the intersection of nano. Um, and please let us know by unmuting yourselves, writing in the chat, or emailing us. We set this up as a regular meeting instead of a webinar so people can unmute themselves and have conversations and really to build a community. Mm. I have two big announcements before I introduce our special speaker for today. First of all, is that we have a new nanotechnology brochure available in nano.gov. This new brochure describes and provides examples of how nano is making an impact and improving everyday lives in sectors that might be of interest to your students. Quantum, AI, consumer electronics, textiles, sports, you name it. There's really cool examples there. You can download it for free on the website, or you can email us and we'll make sure to get physical copies for you. While you're checking that out, there's other brochures in the website that you can also request. Another announcement that I wanted to make is that there's a new and updated nanotechnology timeline on nano.gov. It highlights key inventions and historical events shaping the nanotechnology landscape. I encourage you to share with your students. Let us know if we miss anything important that should be in the timeline, and I would love to hear what reactions your students had to them. And without further ado, let me introduce our speaker for today. Today we have Mr. Hui Pham, teacher at Westminster High School in California. He will share a nanotechnology lesson plan he developed for his biology, maybe AP biology, classroom titled Biotoxicity, Antimicrobial Properties of Silver Nanoparticles. Mr. Pham will also discuss his experience participating in the nanoscience workshop for teachers at the California Nanosystems Institute. I know some of you have already participated there, and maybe some of you are interested to checking that out. Without further ado, let me stop sharing so Mr. Pham can share his screen. It's all you now.
Right there. Everyone can see my screen?
Yes.
All good? All right. Hi, I'm Hui Pham. Look, quick things about me. I'm a UCI alum. I got a biology degree and a chemistry degree, my master's in chem there, and my teaching credential. So everything from UCI, zot zot. Um, I'm currently a teacher at Westminster High School. Have always been there and will hopefully I'll retire as a Westminster Lion. Specifically, I teach bio as my main focus, but I also consult for the chemistry in my school, and I also help with some of the physics. I'm also the STEAM teacher at my school, so I help run a STEAM program with one of my colleagues. We try to have students from our community uh, uh, learn about STEAM. So it's our school is a low socioeconomic, and it's also a high immigrant community. So they might not know, and then their parents might not know what STEAM is, and we try to advocate for them to understand what it is. And it's a great field to go into. So that's my educational background. A couple of little things about me. I like to travel. And apparently right now my thing is to travel to Asia. And probably out of all my travels, my favorite trip was, actually, one of the quotes, sorry, educational, I forgot. One of my educational quotes that I love when my students say, they eventually say, you were right, Mr. Pham. because as teachers we always give them advice, and they're like, no. And then later on, a year or two later, they're like, yeah, you were right. Because my favorite thing to tell the kids is I told you so, you know, um, and their comeback is that yeah, you're right. So that's one of my favorite educational quotes. Like people say it's the aha moment with the kids. No, it's the you were right. But coming back to my other things, I like to travel because it adds to the richness of the education. Not to talk about science, but I like to travel. And my favorite travel thing of all, I've been to Machu Picchu. People think it's that, but it's actually going to here. If you ever know what that is, that's where the North Korea and South Korea borderline is. That's the blue house. There's a special travel that you can actually get there, and you can actually go into that blue house and go to the other side, technically speaking. It's pretty cool. Um, in addition, I'm also I'm now into I've gotten to the stage in my life where I make enough money that I'm okay and I have adult money. So I like to go to concerts. So this past year, I went to if you guys don't know, Twice is doing a world tour. I went to their concert 3 times just for my my new adulting life. Yeah. So that's a little about me. Why nanotechnology? Okay. Again, my training was in biology and chemistry specifically. Um, Nanotechnology was, it's always one of those movie things. And one of the things my colleague actually introduced to me, and how she introduced me was this, free.
Mm.
The biggest, the most important thing, it's free. Free breakfast, free lunch, and most importantly, free supplies. And I forgot the other word, free support um, from an organization at UCLA. It's called CNSI, the California Nanosystems Institute. Uh, and so they provide all these things that were free. That's the hook that pulled me in um, to do it. And the other thing is probably I'm lazy. No, actually, all the science teachers, we work really hard. Setting up labs is very difficult. You have to gather all the supplies, make all the stuff. But this program at UCLA helps a lot of the schools in the area with the supplies, and they also help with the putting things together for us. And they do everything in kits.
If
So
you're ready to sign up for Medicare, find the most up-to-date information at
don't think that's me though. I don't need
Yeah.
Medicare.
If you haven't muted yourself, please go ahead and mute yourself. Thank you.
So my colleagues so actually she's right there. She's actually one of the poster. She's the one in the black. She's the one who introduced me, and I went there and I got hooked. And the food is also great too. So if you're ever in the neighborhood with these workshops, please come. Um, they also help, and one of the nice things is as a teacher, um, trying to keep real world. So CNSI, and I think one of the best things is yes, all that supplies. They uh they give us special tours of their facilities. And this is one of their facilities. This is the one of the electron microscopes. So it makes what we're teaching in the classroom like alive. So that's really cool. And that shout out to Shailene for giving the teachers their special tours afterwards and during too. So I took during this tour, I take pictures of their facilities and stick into my PowerPoint. So I'm not just using a random microscope. It's me at the electron microscopes, or me at the equipment, or me in the next to the clean room. So that's the really cool thing. And I'm pretty sure if you slip Shailene some snacks, she'll give you a tour too, so you can add into your things. And then of course the other thing is NGSS too, right? We all talk about NGSS standard. They also help support with some of those the pedagogy with that. As much as Eileen keeps saying that she's not an expert, I think after this, doing all this thing, she's probably a more expert than a lot of us teachers. And so they provide us the materials that help us connect all those things together. Okay. So again, I got a shout out to the CNSI team for all the stuff that they do. Dr. Tober, Dr. Lake, Dr. Tanemoto, and the recently retired Dr. Marita. I called her Elaine, so I sometimes forget that. So this is not my work. This is the work that they did that I help convey into my classroom. So I stand on their shoulders. So the thing about the CNSI program and the lesson I will talk about is when you first get to it, it is like drinking from a fire hose. Yeah, because they're not here just for the biology teachers, the physics teachers, the chemistry teachers, the earth science. They're here for all the teachers. And of course, NGSS sometimes is a fire hose. There's so many things and you connect in so many ways. So they give a lot of information. So I have here they an example, and I think you guys can see it. Yeah, the links. So their slides is a lot of slides, but it covers a lot of things, and so it's a lot of huge support. Um, so what I do is I don't take everything. I take what is needed for myself. Sorry, we're in eighth period. Um, so they have the teacher manual they support with that, and that also again includes the NGSS standards that helps connect everything. So for other colleagues and for my administration, it's like, how is this helpful? Here it is. They have a student manual again that's modifiable, and I have here a biotoxicity spreadsheet that I developed for my students to talk about science and data analysis. So I'm gonna say this: a lot of teachers when they first get into this, it is like drinking from a fire hose, but after a while, you kind of get used to it and you pull what you need to do. So this lab that I present on their behalf has so much things, but I'm going to come from the perspective of the biology. Okay. So biotoxicity. So again, these slides are not mine. They are CNSI's. They should have the symbol of CNS somewhere in all the slides somewhere, or I might have hidden it by accident. So, and it talks about it's an introduction to the students. And I actually use this as one of my earlier labs because, and I'll explain why. So we always thought about with NGSS phenomenon. So one of the phenomenons or like a demo we talk about is this video. Oops. So I'm not sure play the video, but there's a video of phagocytosis. And in this case, I show this video and then the kids then have think, pair, share where they discuss things, what's going on and let them give whatever possible answer they want. So we do think, pair, share, write on the whiteboards, and I don't actually talk anything about at all. I just let them come up with their own thing. And that's just kind of like, again, a warm up for their lesson. And so biotoxicity, and what I do is I connect this into my lesson when it comes to energy. Okay, so with energy, talk about cellular respiration and photosynthesis. And there is actually a lab that connects with photosynthesis, but I'm going to talk with respiration. There's not that many interesting labs with it or advanced high-tech labs with it. So I connect it with this one. So we actually have already talked about cellular respiration. So this is the kids already know about this. So a week or 2 weeks before, we are talking about cellular respiration. We talk about the mitochondria, the double membrane, the formulas, the energy, and all that whatnot. But there's a huge focus, again, on the formula right down here, okay? And especially on this part right here with the CO2, okay? And then, again, for again, you depending on what it is, I do also have more an advanced class, my AP Bio students. So we do talk, for this one, I'll throw in this slide, we talk about the electron transport chain a little bit more for them. For my regular bio students, I'd be like, hey, you guys remember this picture? Okay, we're moving on. So they're getting another look at this through this lab. So we already went over this. This lab is going to do an application of it. And then, so I do have to teach a little bit of new things. So my kids are in bio, they haven't taken chemistry. So this is where I actually teach and I go, hey, everything's integrated, biochem, physics. I'm going to teach a little bit of chemistry. So we talked about silver, and one of the properties of it is that it can form reactive oxygen species. So I'll talk a little about this. My students, they do know about electrons and unpaired a little tiny bit. So we kind of talk about that, and I can rehash some of that stuff for them through this. And some of them are like, oh yeah, I remember O stands for oxygen. I was like, good, that's good for me. And then we can kind of move on with that. But we do talk about peroxides in one of my previous labs, actually, with catalysis enzymes. So we talk about how, it can be not so great for the cell. Now, so silver in itself has some biotoxicity properties, but typically it's going to be a slow process to sneak through because, well, silver and silver ions, the cells doesn't like to take it in, because it has the enzymes, the channels. It really doesn't want it per se. So it's a very slow process. But so I stopped there just a little bit, kind of pause, have them do, again, I think pair share annotation. Then we kind of skip a little bit, not skip, but then we talk about nano, the special size. So I actually do talk about nano again in another one and more in depth, but here's just more of an introduction where I kind of talk about it on the scale of things. My students actually have already gone through this kind of picture where we did talk about water and glucose with macromolecules and cell sizes. So we did actually talk about this. And here I focus on the nano size, which is somewhere around here with them. And we do that. Again, one of the difficult things for students is scale, size and scale. So, again, what's the scale of $1 million versus $1 billion? And we do a little discussion about that here. So, yeah. And then I point out a couple things about that silver actually has different types of silver, powder, nanoparticles, and silver ions. And there's a little bit about that. Now, in the full-on CNSI, what they train is because they do all the NGSS, they actually have part of the one part of the experiment is to synthesize silver nanoparticles. I don't do that because I don't have the time, and these biology kids, they don't. But so I synthesize it with my aides. So my aides are they've already taken chemistry, so we synthesize it separately. Just kind of give you guys a heads up on that. So then we talk about, again, silver. And one of the big themes in biology, again, is surface area. I think if you teach biology, surface area is a big theme. So we kind of go through this. I do have them draw this out and try to tie in the math aspect with this, with surface area. So we do have a small talk about surface area and the difference in like macro versus very small atomic versus here, which is the in between, the nano size. And what's the difference? So surface area to ratio for silver is much higher at the nanoscale. They kind of get that. I'm not going to say like I'm like a miracle worker, but some of them do get that enough. Some of them, I'll be honest, will never get that. It's because some of the kids' math levels it's a range. And then a lot of students actually have heard about Trojan horse. So we actually do a quick discussion on the Trojan horse before I go over the slide. We talk about the movies. What's that movie? Achilles. Have you ever watched Achilles? So they kind of know about that and the War of Troy. So we kind of talk about that. And here is probably the biggest slide of all that I need them to know about the properties of nano. That silver nanoparticles, because of their surface area to volume ratio, they can be mistaken as food. They're that size, so the cells can somehow it can sneak in. And again, the toxicity is higher when normalized due to the mass. Again, that surface area to volume ratio. So this, out of all the slides, I do want them to realize. So I will stop here. Let's talk about this. Again, not every student, but they do understand. Oh, food like what? And they kind of remember, some of the students will remember, wait a minute, that picture that we had, which is right here, somewhere, somewhere around here. Yeah, there you go. They kind of do remember this. And then, so as long as they get this slide, I'm actually good. And then we talk about, okay, at nano, there are different properties. And one of the properties, and this one, a lot of them have a trouble understanding, not into another lab that we do. It's the light interaction. So this is the fire hose part for the kids, that particles and nanoscale interact with light differently. Be honest, most of the kids don't understand this fully. But it actually is a good introduction for another lab that I talk about later on with it. But I said, what's color silver? And they all know it's, a silver ring is silver. And then atomically, we do know because we do like salt and it dissolves, it's white, and now it's, oh my gosh, it's transparent. They kind of understand that. And so I said, hey, here's a silver salt. What happens? They know that. But the nanoscale, because light interacts with differently, silver is a different color. And it's the idea is this, hey, you guys, if it's yellow, it looks like kind of like pea, silver, but it's a nanoscale. That's all I need to tell them to know. Here's regular silver, macro. They all know that. And then atomically, salts, it's transparent. And then here. And then I go, here you go, guys. I made it for you guys. So how's this done? So what's all this stuff to do? We then do a lab that relates to biotoxicity, where this lab is actually pretty simple. The hardest part is the nanoparticles, synthesizing it, but UCLA made it very easy to synthesize. to a degree. So this lab that we do is very simple. I think it's a modification of another one, which you have respiration. You just need a flask, sugar, which anybody can buy, our yeast, and then warm water. Yeah, the key is warm water. I learned that cool water makes it very slow, and my period's only 50 minutes, so I got to make it a little. So it has to be warm water, and then a manometer. So this is the one that UCLA puts in their box for us. It's made out of cardboard with tubing, colored water. So this is the one they gave us. Since I do this lab every year, I need a more permanent one. So instead of cardboard, I take these things and I built it on this, and I can just stack them somewhere. So our tubes that are actually taped on, so they're permanent for me. And I teach the kids how to use the manometer a day before. Okay. And so it's respiration. It's because respiration is related to the yeast being alive, right? And if they're alive, essentially, they will do respiration. You produce what? CO2. If they're dead, they don't do respiration. What do? You don't produce CO2. So those are the two extremes. And then we'll talk about everything in between. So again, apologies if I am going fast for this. I don't know my timing. So we do have 4 different samples. So the class is split up into, I have 8 groups. So there are 2 groups that do each of the different types of flasks, and they'll gather data. So we have our control here. We have our silver nanoparticles, plus all this. We have our bulk silver. And I know silver is expensive nowadays, but thank goodness, again, UCLA supplies it for me, so I don't have to buy it. And then the same thing with the silver ions, where they give me silver salts. And I just make 4 big flasks where the kids will then pour into smaller ones, and then they'll do the experiments. And again, the first trial, they always mess up. It doesn't matter which of the situations. So I have them do multiple trials of each. And the idea is then they do the lab. And to be honest, individually, they'll get some data. And they're like, I don't know what's supposed to happen. And I think that's one of the valuable things. And I go, that's okay. Because guess what? You're only seeing one data point. Oh, and if I didn't preface this is actually a 3-day thing, because we also have another day where we do analysis. So they do the lab. Again, they add all this, do this, put it in the manometer. They stick it in the manometer right here. They collect their data. And this is what the data kind of looks like. So this is the data from UCLA for one of our labs. And this is what it tends to look like if it's perfect, right? And as you can see here, the control, because there's nothing in there to kill the yeast, it will have the highest respiration. The height of the manometer moving, carbon dioxide produced is very high. The silver powder, also very high, because again, cells don't take it in. The silver nitrate is a little biotoxic, and then the nanoparticles are the most. This is what the data should look like. I don't show the kids this though, not that first day. What I'd like to do again is I put it into this spreadsheet. So I built a spreadsheet where the kids do 4 trials, and they dump their data in here. Again, they don't know anything, but again, we're able to, especially with this lab, we can do a lot of data analysis. And to be honest, I don't know if a lot of teachers do data analysis because it takes a lot of work, it takes a lot of time. You got to go over it. But I like this one because it does produce so much data. It's easy to replicate. Each trial actually is not that long. So within one period, the kids can do all 4 trials. So this is the blank one. And then the kids put it in, and they'll get data that looks like this. Okay. So this is a sample, one of my sample data. And data to me is data. Doesn't matter. I actually do like it when the kids mess up, because then we can poke fun of it. No, we can talk about outliers. We can talk about what went wrong. And some of the kids were like, oh, you're right. That is weird. Why 3 of my trials look the same, and then the 4th one was just off? So this one is what the student data does look like. Here in this one right here, you'll kind of notice that the nanoparticles isn't actually the lowest. It's the nitrate, right? And that's okay because I do have usually I have about 3 periods that do this. So I have 3 periods of data. And I cross my fingers that at least one of the periods gets it right. And generally, when you have 3 periods, 1 period will get it right. And if not, my 2019 will always save me. The one class that does perfectly everything. And I also have then pop this data in as something that the kids can look at. But we use it. We first look at this, and we can see trends. And again, outliers. They talk to each other. Again, this is a separate day where they talk to each other, and they're like, "Okay, this is cool," and what's going on. And then the next day, they'll do some of the post lab questions individually, what their initial analysis is, and then the next day, so that was homework. The next day in class, I'll talk about this as a whole. We'll look at each period. Well, actually, they'll look at their own periods, discuss. Then they have the other periods. I'll pop in, and they'll discuss those. And then I go, hey, you guys, here's my perfect data. Now do a comparison of all that. And a lot of the kids will see, okay, general trends are there. Most of it, again, usually the nanoparticles is the lowest, but sometimes they'll get something off. One of the favorite things is when the none actually is lower. Then they'll be like, what's going on? And we discuss this. So there's that scientific investigation skills, but then the nano is there. The investigation skills is always in the background of everything. And so here, it's a great introduction. And this is one of my labs I do in the first 2 months to nanoparticles. Again, they still don't know the whole idea of nano, but they kind of see that, hey, it has special properties. And we also do a reading on, after the lab, I send them a separate reading. For one of my FRQs, I give them an article, and they'll read about a silver being used as for antimicrobial properties. And that's through one of the articles I think one of the UCLA grad students showed me, I think, or it's one of I pulled up. So yeah, so that's the general one. Again, I out of the whole fire hose where the UCLA teaches everything, they also teach the synthesis. I don't do the synthesis. So the whole lab, they do the synthesis. And then what you're supposed to do is use the synthesis in the next day to do that. But again, I have sophomores and they're sophomoric, which means they're mature. And they're still learning how to do labs. So there's also that too, that if I ever share this stuff, they also show the synthesis. It's not too bad. My aides can help me. They've taken chemistry. They kind of know how to do that with that. But I do some follow-ups with this lab later on. So these are a series of labs. So I don't just do this. I try to incorporate into my biology class too. There's a measurement at the nanoscale and nanostructures that follows up. And that's where we use lasers and diffraction gratings to do nano measurements. And then we also do a nano chocolate to talk about nanostructures, because nanostructures do that. That's a really great lab. I actually just did it a couple weeks ago with my students where they made the chocolates, and they love that. And then again, tying in, there's another lab that I do. It's called dye-sensitized solar cells, where we use titanium oxide as the nanoporous medium to soak in anthocyanin dyes, and we make a solar cell. And they go outside and they measure it in the sun. And I also learned some kids don't know where the sun, if it's cloudy, they don't know the sun rises. And then, yeah, it's really funny because it's like 8 o'clock in the morning. They're holding it straight up, thinking the sun is like, no, you guys. So we all have those kids, right? They're like, oh yeah, the sun's over there, you guys. So we do dye sensitize, and it's pretty cool because it's using raspberries to make a solar cell. Again, biodegradable, very cheap, actually pretty easy to do, to be honest. If UCLA just gave me the recipe, I can get it. The hardest thing I think for me to order would be the glass, the conductive glasses. But everything else, raspberry I get the store, pencils, titanium oxide powder I can get, and then acetic acid I can get. But they give me so much. Sorry, Shani, I have so much. Next time I'm just going to order only the glass slides. So that's another one. And again, it ties in with photosynthesis, where the kids do that. We also do a biopolymer. I also do biopolymer, and that one I flip throughout the year where I have time. Sometimes I do, I don't, I just move it around. We measure Young's modulus with that one. Interesting story. I actually had biopolymers as my lab this week. But if you don't know where Westminster High School is, we're next to Garden Grove. My students actually got evacuated from that Garden Grove chemical fire. Yeah. So this lab was cut out. Sorry, Shailene. I ordered material, but I had to cut out because I had to have to help my kids catch up. So, but it was a teachable moment because I could teach my kids about MMA and pressure and Belv and all that stuff. Yeah. My students did, and some of my colleagues actually were evacuated. My school is actually on the other side of the border. So the street where the evacuation zone, and then there's my school. There's another school neighboring to us. They were the Red Cross shelter. So I didn't do that lab this year, but I do that lab, and that one's pretty flexible. And then I also do a super hydrophobic, where we synthesize a super hydrophobic using nano material to roughen the edges so that we can make a super hydrophobic, which is kind of, again, the lotus leaf effect. And that's actually my lab I'm doing as my end of the year capstone as the final lab for them. A couple other things I just want to talk. We did photolithography. So I don't teach physics, but I consult. So I went in and I consulted and I taught them how to do photolithography. And they just did that lab. And then my chemistry does water purification lab too. So there's a couple of the follow-up labs. So the first four, actually, they all connect together and I can make a story out of them. And then these two there's two other labs that the teachers at my school are also trained in doing. So we do these two also. So yeah, that's my thing. If you have any questions about that, it's a very good, for me, good intro at the beginning of the year lab that I've always had trouble finding a respiration lab besides, here's yeast, make it balloon, and you get carbon dioxide. But I wanted to tie in a little bit more with some application, which was antimicrobials. That was my application because a lot of my kids are into healthcare, so we can talk about that, antimicrobial and things like that. So yeah, talk a little about my CNIS experience. Again, none of this is mine. I just stand on their shoulders. My CNIS experience. This is the type of PD that teachers want. A lot of times my administration says, you got to go to this PD. I was like, oh my God. But these are held on Saturdays at UCLA, and every teacher there wants to be there. Because they're giving up their Saturday, but they want to be there. And UCLA really respects us, and they make it engaging. We have a great network of teachers. I mean, where we then, network with teachers. I'm from Orange County. UCLA is in LA County. It's an hour drive from me-ish with traffic, but we have teachers drive 2 hours there just to kind of go learn and network with each other. Again, there's free food, so that's also a bonus. We also exchange science pedagogy. I do bring some new teachers and other new teachers, and then they learn not just nanoscience, but in our conversation at the beginning, we are also learning about others that's not specifically nanoscience, but just science in general. So there's a lot of science pedagogy. So yes, the center is nanoscience, but nanosystems, but they have grad students there from all different type of disciplines. And then we get to troubleshoot with the labs. So, I might do one of the labs and I have a problem and another teacher did it too. And we came to the next workshop and we're like, hey, you did this. And we're like, yeah, and what's going on? And we can share and troubleshoot and make it better. And then we also asked the UCLA grad students and Shailene, how do you troubleshoot this? And we all troubleshoot together. And then again, exchange of knowledge for new demos for everything, new pedagogy, new science experiments and demonstrations, especially a lot of the younger teachers, definitely. Again, lifelong learning. We're science. We're always keeping up to date. One of the great things is Shailene brings in guest speakers for lunch, and they're talking about the latest and greatest what's going on to keep us on edge. We talked about how nanotech was used for a catheter synthesis. We did a supercapacitor where they're talking about batteries and all that. So these are again, UCLA is one of the top. I'm not gonna, it's one of the top. Sorry, the other schools, but it's one of the top public schools in the nation, and they bring in the best for us to learn from. So yeah, with all these real world science that I can bring to the students, and then of course to socialize with other teachers, pedagogy, and then we also vent with each other sometimes. Sorry, that's some of the biggest things about our kids, about this, that, and everything else too. And it's a great venue network. I work with a lot of other teacher things too. And again, when there's a PD that the teachers come on their own volition, it's going to be great. And then we have a great support from the CNSI staff plus the grad students who come and talk. And those grad students volunteer their times. Okay. A couple things that I also do outside, or actually, I don't say outside, that does connect this is I also work with the Orange County Science Engineering Fair. And some of the experiments that my students do actually are inspired for that. We've actually had an antimicrobial one with not silver nanoparticles, but they synthesized copper nanoparticles as a cheaper alternative. And they did it, and they submitted into the research for the county science fair, which goes to the state science fair and the international science fair. Specifically, I actually work at my school with other Title I students in it's called the Mentor Match program. So within the Orange County Science Fair, we actually have a special program. We work with Title I students, the students who would never participate in science research. And I'm one of the first set of teachers, so that we give those students the chance from middle school and high school. These are underrepresented students in neighborhoods that you'd be surprised there would be kids doing science fair at all. And I'm proud to say that we've supported 200 students doing that in Orange County. And again, some of the kids are inspired by the labs. They might do a modification of that. I had a student actually do a water filtration. We did a water filtration, the nano one, and then they said, hey, they read a research paper and they did another type of nano filtration, and they were recognized at the county and the state for that research. So it inspired them. I also work with another program called Chief Science Officers. So we do science and then we advocate for science. So my students, with all the knowledge from everything that they do, this is an international or national/international. They go into the community and they teach science. I had students go to Washington, D.C. to SLECOP. I don't know if anyone's participated. It's like the National STEAM Conference, and they were the only students to speak. We also go to the elementary schools and we do workshops for them, or the kids do. And then we host STEAM fairs. We do a lot of CTE and career building too. That's another program I work with. Again, and then these kids take their experiences. We also do competitions. Again, shout out to UCLA. That's my students in front of the California Nanosystems Institute. They do a Shark Tank where you take a nano science and you make a product and you pitch it. So they pitch it to judges from UCLA. They get mentorship from UCLA too to do this, and it's on a nano product. I think this team right here did a nano derm, and then the team this year did a nailed it, which was a nano inspired nail glue for the nail extensions. And then I also do other things too. Besides that, we build the go-karts, electric go-karts. So I also work with those type of competitions. I also host conferences for my students, again, in their advocacy. They host conferences. This is a Girls in STEM. I just invite the middle schools, and my students run the conference, and they show everybody the girls can do STEM or STEAM. We also host Hispanic and STEAM because that's one of our other underrepresented groups where my students host it. I just sit in the back. And then Shailene also sponsors it with amazing t-shirts for the students to understand that everybody can do science. I also do Explore Vision. So some of the science, so it's Explore Vision, if you know, it's from the National Teachers Science Association, where the kids think 5-10 years in the future, some science that can be wow there. And I actually do have students who, again, are inspired by nanoscience, not nanotech from Marvel, but from nanoscience. And they come up with a science idea and they submit into Explore Vision, their project. and they get recognized there too. So they're doing a research, like a thesis paper on it. And nanoscience, like nanofibrils and nano antimicrobials, that's actually they've won with those ideas. So this is one of the products, the examples, and again, it's very open to everybody, and it opens my students to that collegiate level things. And then lastly, probably one of the reasons why I'm so winded today, I also work with my school's farm. I have we're in an urban area, but we have a school farm. We have an orchard. We also have a giant field. If you ever come by my neighborhood, we're picking strawberries. And then I'm actually here. My students are actually outside over there. They're actually taking care of the bees right now. So that's why I was a little slightly late. Um, Interesting thing, one of the kids' project was to find a nano product medicine to help bees colonies. That's one of their research ideas. So again, inspired by nano. And there's actually a research on a nano medicine for bees that there was some PIs working to help against the what is it, mites. So anyways, that's a lot about me. Fire hose. If you have any questions, feel free to ask. I didn't know how long I'm supposed to talk for. I didn't know it was a whole hour or the 30.
Virtual round of applause. Thank you so much. What don't you do? I feel like you're involved in everything. And thank you for everything you do.
Yeah, that's
I think with that, let's open up for questions. While people decide to unmute themselves, maybe I'll take the prerogative of the first question. Going back to the lesson plan, I'm
Mhm.
curious, what was the first wow factor for your students? What was the first thing in the lesson plan that they're like, oh wow, that's cool.
Oh, it's the manometer when it starts bubbling, and for like the uh the no control, it just bubbles out. And then that's one wow factor. And then you have the opposite, in which for the nanoparticles, like, hey, Mr. Pham, it's not moving. Like, what's going on? I'm like, well, think about it. And then sometimes it's like that clock going, and I'm like, oh my God, it makes so much sense. Well,
There's
duh, the
that.
nanoparticles don't, they kill them. They shouldn't make any carbon dioxide. So they're like, oh, yeah. So it's just fun seeing again the bubbling, and
Hmm.
it's the rest, it's like the macro aspect, but
Hmm.
there's the micro that we do discuss, and they're like, oh, it makes sense because each of them, to be honest, if we had time, they did, each group did all four, it'd been amazing. But time constraints and of course supply constraint, each group can't do all four. But
Hmm.
they each individually, and they're like, oh. So that group that actually does the nanoparticle ones, they actually get a little bit more learning. They're like, oh, that's why we're not getting any bubbles or any gas movement.
Okay.
Um,
Very cool.
Probably the other one is it looks kind of like pee. Is that silver? Yeah, it looks kind of like pee. Did you pee in it, Mr. Pham? I was like, yeah, I peed in it. Don't drink it, guys. I peed in it. Just don't drink it. No, we go, it's nanoparticles. They do have that color. It actually gets reinforced during the nanostructures, because when we do the light splitting, and they're like, oh, color, it splits into the colors. Oh, so there is color at the nanoscale. It works differently.
Well, if they like color, there's some really cool experiments with gold and gold nanoparticles. The
Yes.
students always like that.
Yes, there is a lab that Shailene teaches about that, plasmonics. Yeah.
Any other questions from the audience? Yara, please go ahead.
Hi, everyone. Thank you so much for this beautiful presentation. Honestly, it was very impressive. My question is regarding something you mentioned something in the follow-ups regarding solar cells and raspberries. So I'm really curious what that is. Could you please provide some more details on that?
So the 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 nano part comes from this. So we take a piece of glass that's conductive, because the solar cell, you're generating electricity, right? And then you coat it. If you put anthocyanins, which is a dye, it can absorb light. You put it on there, you get only so much because the glass is like flat. But if you put titanium oxide on it and you coat it, and there's like a cooking process, you coat it. The titanium oxide does nanoporous holes. So it increases surface area. So what happens is when you squeeze raspberry, you get the juice. And in the juice, there's anthocyanins. You take um, the nano-coated, the titanium oxide nano-coated glass, you soak it in there. The anthocyanin soaks into the solar cells, and so you have a huge surface area that can absorb a lot of the anthocyanins. So when you take the solar cell out, the sunlight hits the anthocyanins, and then they can do this reaction that can generate a redox reaction that can generate electricity. We don't measure it through a light bulb, although one of my kids really wanted to put everything into series to do it, and he actually did do it for fun. During the break, he took everybody's solar cell and he started putting it in. But with a multimeter, they can measure the numerical change for that. And the kids really like angling it at different angles in the sun to kind of get the numbers to go up and down.
Okay.
So it's again, I think if Shailene ever does a virtual thing, if you're not in the area, that's definitely easy to get the material, to be honest. I think and it's actually one of the easiest one for that particular app. And actually, if I think about it, I should have presented on that one. My student teacher went to the training one year, the first year, and I said, "This is your end of the year to pass as my student teacher. Take this, turn it into a lab that the kids can understand and teach it." So I use his lesson. So my student teacher taught it, and he followed everything. And it was like it's probably one of the favorite labs for the kids. But again, maybe on another workshop, Shailene can con Joshua to presenting about it.
We can definitely pressure Shailene to do it too.
And she can, I definitely all the materials is easily purchasable. It's cheap.
Thank you so much. It's really impressive, honestly.
Mhm.
I'm very happy to hear about it.
Yeah.
We have a question in the chat. Is there a student version of the lab activity that could be shared with us?
Well, that is up to my boss because I don't know how proprietary information it is. So I'm gonna let her answer that.
Also, I'll let her respond, but also someone else, Marielle replied that there's a video online. But Shailene, please go ahead.
Yeah, so again, I have to defer to my boss even. So Professor Tolbert, who runs our program, she's our faculty director, and she very much likes for the teachers to be trained by us in order for us to give out the materials. It's just about trying to make sure that the science is explained correctly. But I feel like the dye sensitized solar cells, again, like there's a lot of stuff out there about that. I don't know if in your slides here, you had the presentation and the teacher manual, student manual. If that links to something, you know,
It links to, oh, it links to
I,
it links to my stuff. Oh,
right,
it
your
links to
versions of stuff.
my version of stuff for biotoxicity.
Yeah. So not, I'm not gonna stop that link from being active. So if people want to click through, because I know these slides will be shared after, can't stop you. Again, all of this is fairly reasonable. We give the kits out for free because a lot of our teachers come from Title I schools that do not have money for supplies, and we don't want our teachers spending their own money to buy these materials. So that's kind of our mechanism of gatekeeping is that we give our supplies out for free, but all of this is sodium citrate and silver nitrate and silver powder, and none of that is like anything particularly difficult to get. It's yeast and sugar, and
Yeah.
we have a manila folder that we turn into a manometer. Like it's not anything that's very difficult to get yourself. So if we is sharing these resources and you want to build your own stuff from it, I am all for that.
I mean, we're science educators. More science education is always better for society.
Right.
We're not gatekeeping.
Right, so I put the link, hi, this is Mariel. I put the link to the dye sensitized solar cell kit that you can purchase online, and that has all the instructions in it. So you can actually make those without any kind of issue. But I would like to, I am interested in the biotoxicity lesson. So
Mhm.
I'm looking forward to diving into that.
Thank you. If if there's any other questions, please unmute yourself. I'm gonna read another one from the chat. So Brandon Prof asks, what is your favorite example of practical applications of nano? And when your students inevitably ask, and how do you respond to how do we want to kill yeast more quickly?
So there's two. Wait, I gotta look at the chat
It's
again.
two questions, yes.
Yeah, let me try to just look at the chat myself. What was the first half? First question? Oh,
What are
well,
your favorite examples of practical applications of nano?
oh, it's gonna come to this lab right here, which also connects. So it's this one right here. The best one is going to be the nanostructures, because they make the chocolate, and that's something they actually can physically do. But it's kind of akin to a photolithography where you're using nano to make microchips, and they understand the importance of electronics. For the physics kids, when they did this, it's like, again, we talk about all this photolithography and lithography stuff. Um, it's out there, but they're actually doing it. And my colleague who actually did it with his regular physics students, Daniel, it was just like, oh my gosh, I'm actually they're holding something that looks, and they're like, oh, you know. And then for my students, when they do the imprint lithography for the chocolate, and they see that it's rainbow colored, they go, that is a really cool thing to do.
Mm.
And in my lab, I actually say, you're gonna do this. I make little kits for them with the diffraction gratings, and I give them so again, Shailene gives me the chocolates and the stuff, and I put it into little kits. They actually do that portion of the lab at home, and then they have to show their parents, and their parents say, "You've done something pretty cool." And the fact that, again, it's imprint lithography using the nanostructures, um, that gives the kids a, they really like, now they kind of understand what I said when it comes to like light interacts with nano scale things differently, because they can see, oh, no rainbow.
Mm-hmm.
The only difference is the nanos, you can't see it, but they know that there's a structure, because they can also look at the diffraction grating through the lights, and they love that. Everybody loves pretty lights that separates.
Mm-hmm.
That's the favorite thing. And again, sometimes the kids don't connect it right away, but in the end, like after 2, 3 things, they start connecting things, and they start making the aha moment. And I'll be honest, most of my sophomores, they do get it. And then at the end of around the junior year, when they start doing more stuff, they're like, oh my gosh, that makes so much sense. You know, that you were right, Mr. Pham. That is cool. What was the other half of the question again?
Oh, the other half of the question is, how do you respond to the question, why do we want to kill yeast more quickly?
Oh, this question was, yeast is the model organism. It models the organism for microbes. That's what it is. I've also, and you said, yeah, don't you guys ever get yeast infections? There we go. So that's something that we talk about. But they do know it is a model. I'm using yeast as the model organism.
Mm.
And I tell them in my biology class, yeast is model for a lot of different microorganisms. It's safe and it's cheap. When my kids do science fair for research, we have to do like, it's called SRC or IRB type of things. And I said, when using yeast, we're not going to use that E. coli. I don't want you guys to deal with it. And if it equivalates enough, it's perfect.
Mm-hmm.
So they understand. I do mention again, it's yeast is a microorganism model for us. And so you kill it quicker, infections, it grows, and you can take out infections.
Yeah. Okay, we have time for one more question, comment, or concern. Anyone wants to speak up?
Uh, if no one else is, I'm gonna make a pitch. If you ever, steal the UCLA website, I did not put it in here, but if you ever can, get a PD to come on that Saturday, it's free and it's lazy. They provide, there's a lot of huge support on it, and I don't know if any other places have that type of PD, but it's great for all types of science.
Thank you. Thank you for saying that. And I think it's a great shout out to a great program. I've heard so many people are alum from this program, and I love that it keeps growing. There's other similar programs in different universities around the country. So if you're ever very excited to do a PD like this, but you seem to not find something close to you, please email us and we'll be able to point you in the right direction. And with that, another round of applause to Hui. Thank you so much for everything you do and for sharing it with the group. And I hope to see the rest of you in our next NanoEd forum, probably in September. Have a great summer. Keep in touch, everyone. And as a reminder, everything we share today will go in the recap email, and I have that Google Sheet with all the resources from today and all past sessions. Thank you again, everyone. Bye-bye.